Hello students, welcome to EPG Pathshala. I am Dr. Zeenat Iqbal from Department of Pharmaceutics, Faculty of Pharmacy, Jamia Hamdard. Today we are going to discuss about the types of control release systems in this module. This comes under the paper NDDS Part 1. So students, let us see what we are going to learn from this module. The first aspect of the module would dwell upon the drug release patterns of the controlled drug delivery. We will remain limited to the zero-order release profiles of such systems and the first-order release profile. We'll try to get into the details how it is done while we are making or manufacturing a controlled drug delivery device. Then we will spend some time on just elucidating the different types of control release systems. We will limit ourselves to the oral control systems, parenteral control release system, transdermal drug delivery system, ophthalmic drug delivery systems, and intravaginal and intrauterine drug delivery systems. My dear students, the primary focus of the controlled drug delivery systems is usually on two important aspects. The first thing is that it has got a goal of reducing the dose, and the second one is that it has a goal of reducing the dosing frequency. In order to meet this important goal, which primarily will result in the patient compliance, the release pattern of the drug from the controlled drug delivery system device is important to be understood. If a drug is released at this particular rate, it will primarily be suited for the oral release, oral drug delivery route. If it is probably needed that it should release its drug over a period of 12 hours, then we will have to have a different designing parameter. If it is expected that the drug has to be released maybe for a period of more than 10 days or 12 days or 13 days, then the pattern of the release has to be pre-decided. Before we move into the actual designing of the controlled drug delivery system, we usually have to plan or make a blueprint of that. So we need to understand what would be the drug release mechanism. For this, what we do, we need to understand what are the various patterns. Although there could be some complex patterns as well, but we will limit ourselves for the understanding of the zero-order release pattern and the first-order release patterns, which are the commonest type of drug releases. Drug release pattern of controlled drug delivery. It is absolutely understandable that the final behavior and success of a controlled drug delivery system will primarily depend upon the drug release pattern which it exhibits. The first exposure to an aqueous fluid, the controlled drug delivery system, which primarily consists of hydrophilic matrices, will take up the water and the polymer starts hydrating to form a gel layer. An initial burst of the soluble drug may occur due to surface leaching when a matrix containing a swellable glassy polymer comes in contact with an aqueous medium. There is an abrupt change from a glassy to a rubbery state which is associated with swelling process. With time, water infiltrates deep into the case, increasing the thickness of the gel layer. This is very clearly indicated in the figure in front of your screens. Concomitantly, the outer layer becomes fully hydrated and starts dissolving or eroding. When water reaches the center of the system and the concentration of the drug falls below the solubility level or value, the release rate of the drug begins to reduce. At the same time, an increase in thickness of the barrier layer, with time increasing the diffusion path length, reducing the rate of drug release. Drug release kinetic associated with these gel layer dynamic range initially from Fickian to anomalous or non-Fickian and subsequently from quasi-constant near zero order to constant. In general, two major factors control the drug release from the swelling controlled matrix system. They include many processes given below. The first one is the rate of aqueous medium infiltration into the matrix, followed by a relaxation process, that is hydration, gelation or swelling, and the rate of matrix erosion. As a result of these simultaneous processes, two fronts are evident: a swelling front where the polymer gets hydrated and an eroding front. The distance between these two fronts are called diffusion layer thickness. Diffusion layer thickness depends on the selective rate at which the swelling and eroding fronts move in relation to each other. If the polymer gets slowly, solvent can penetrate deep into the glassy matrix, thus dissolving the drug. Therefore, the gel layer thickness and its stability control the drug release. For example, swelling of the HPMC matrix tablet was found to be higher for a higher molecular weight. They attributed this to the large hydrodynamic volume occupied by higher molecular weight chain when hydrated. As the polymer chain becomes more hydrated and the gel becomes more dilute, the disentanglement concentration may be reached, that is, the critical polymer concentration below which the polymer chain disentangle and detach from the gel matrix. Drug release has been an important topic in the field of drug delivery for decades. With advancements in material design and engineering, novel materials with increasing complexity and more functions have been introduced into the development of drug delivery devices and systems. Both naturally derived and synthetic macromolecules are extensively used in controlled drug release to maximize bioefficacy, facilitate clinical applicability, and improve the quality of life, just to name a few. Drug release primarily refers to the process in which drug solutes migrates from the initial position in the polymeric system to the polymer's outer surface and then into the release medium. This seemingly simple process is affected by multiple complex factors, which will be understood in just a little bit of time. My dear students, there are a large number of factors which actually will influence the drug release from the controlled drug delivery system. These factors can be headed as the factors which are governed by the material matrix, then the factors which are the reason from the factors which are from because of the release medium into which the drug is released, and the third category are the drug related factors. Let us take them one by one. The first category of the factors are concerned with the material matrix. When I say material matrix, I intend to put up the point that it is the polymer or it is that material of which the controlled drug delivery system is designed. What are the factors? The composition of that material, the structure of that material, and the swelling and the degradation of that material would influence the drug release from such systems. Then the second category of the factors include the ones which are related to the release medium. So we need to understand that the pH, the temperature, the ionic strength, the presence of or absence of enzymes in the release medium will definitely influence the drug release. Then thirdly, the drug component related factors. The first one could be the solubility, the stability, the charges, and its interaction with the matrix. My dear students, the figure in front of you is just a position of the drug release pattern of controlled drug delivery system and a conventional drug delivery system. You have the plasma drug concentration on the y-axis, while on the x-axis you have the time component. The dotted lines primarily, the lower one shows the minimum effective concentration, while the top one shows the maximum toxicity concentration. The red colored figure or the graph line shows the release from the conventional dosage form, the first peak and valley, the second peak and valley, and the third peak and valley, while the blue colored line is giving the sustained or the controlled release action, and sometimes referred to as the modified release dosage form in a plateau form. There are a number of kinetic models which describe the overall release of drug from the dosage forms. Because qualitative and quantitative changes in a formulation may alter the drug release and in vivo performance, developing tools that facilitate product development by reducing the necessity of biostudies is always desirable. In this regard, the use of in vitro drug dissolution data to predict in vivo bioperformance can be considered as the rational development of controlled release of formulations. The methods of approach to investigate the kinetics of drug release for controlled release formulations can be classified as the statistical methods, which is also referred to as the exploratory data analysis method, and the multivariate analysis of variance method called as MANOVA. Then you have the model dependent methods, which will designate the release as zero order, first order, Higuchi's, Korsmeyer-Papas, Hickson-Crowell model, etc. And finally, we have the model independent methods which probably will assess in terms of F1 and F2, referred to as the difference factor and the similarity factor. Under the zero order release profile, the drug dissolution process from the dosage forms takes place at a constant rate and independent of the concentration of the drug. Zero order release is primarily predicted as Qt minus Q naught is equal to K naught T, where Qt is the amount of drug dissolved in time T and Q naught is the initial amount of drug concentration in the solution, and K naught T is equal to the zero order rate constant. The primary applications of the zero order release profile is for pharmaceutical dosage forms which show modified release, which might include the transdermal systems, matrix tablets for drugs which are soluble and can be delivered in this system. Students, let us just very quickly go through that how we can probably assess the zero order release profile. We have just explained that what is zero order, that it is concentration independent. Let us see that when this cumulative percentage drug release data is plotted against the time and we obtain a plot which is linear, then we can ascertain that it is following a zero order kinetics and the slope is equal to K naught. This model primarily represents an ideal release profile in order to achieve the prolonged pharmacological action. But practically speaking, it is absolutely very difficult to get to the pure zero order release profile in case of the products based on the controlled drug delivery. We will subsequently now discuss the first order release profile. It is a linear kinetic process in which the release rate increases linearly with increase in the concentration of the drug, and hence the process is concentration dependent. A first order release would be predicted as follows: log Qt is equal to log Q naught minus K1T divided by 2.303, where Qt is equal to the amount of drug released in time T, Q naught is the initial amount of drug concentration in the solution, and K1T is equal to the first order rate constant. The application of the first order release profile are well extended to the transdermal systems, matrix tablets with low soluble drugs in coated forms, and osmotic systems per se. Students, let us visualize that what will happen if we plot a log cumulative percentage drug release and time on the y-axis and x-axis respectively. The graph which we will obtain, or which is seen in front of you on your screens, is primarily a profile equivalent to first order release. The data was taken to be in the log form of the cumulative percentage release. The x-axis had the time factors and the constant k can be obtained multiplying the slope values. When the data yields a straight line indicating that the release follows the first order kinetics. Students, I think we are probably able to appreciate the fact that the controlled drug delivery systems is not limited to a particular route. It can be explored on different types of route requirement. If we just try to recapitulate what is a controlled drug delivery system, we can say it is a delivery system which releases one more or more drugs in a continuous fashion in a predetermined release pattern. Mind you, the word predetermined release pattern is important here for a particular period of time, either locally or systemically, to a specified target organ. Depending upon the route of administration, we can be very readily able to design the oral control release system, the parenteral control release system, the transdermal control release system, the ophthalmic drug delivery systems, and also the intravaginal and intrauterine drug delivery system. Subsequently, we will take each one of them and try to understand the nuances of the same. The role of ideal drug delivery system is to provide proper amount of drug at regular time interval and at right site of action to maintain therapeutic range of drug in blood plasma. Controlled release dosage form is a dosage form that releases one or more drugs continuously in predetermined pattern for a fixed period of time, either systemically or locally to specified target organ. One of the most importantly exploited route is the oral drug delivery, and for that there is a lot of attention paid on development of the oral control release drug delivery system. It offers a lot of flexibility in designing of dosage forms, and there can be various approaches which probably are primarily used for making such dosage forms. The approaches are diffusion sustained control systems, dissolution based systems, ion exchange based systems, osmotic pressure based systems, pH independent formulations, and sometimes the altered density formulations can also be formulated. The controlled drug delivery systems focused on the oral delivery or the delivery through the oral route can be of two types: the controlled release and the delayed release. The controlled release systems control the site and rate of drug release, and under this we can have the sustained release, which maintains the drug concentration within therapeutic window for maximum period of time. The extended release system reduces the dose frequencies and maintain a constant plasma concentration, while the prolonged release system increases the duration of action of a drug. Subsequent to the controlled release, we have the delayed release oral systems. In this case, the drug is released after a lag time followed by administration, which is achieved by the incorporation of a special coat. A typical example can be a formulation tablet which is enterically coated and referred to as an enteric coated control release drug delivery system. The oral controlled drug delivery systems usually have got very conspicuous advantages. The first one is the reduced dosing frequency. Then we have the second major advantage as the dose reduction. Then definitely these two parameters are going to increase the patient compliance. Then the other advantages, we can have probably the predetermined and constant plasma drug concentration for a prolonged duration of time. We of course can reduce the toxicity due to overdose, and this can also lead in reduction in fluctuation of peak valley concentration. And of course, one major advantage is the avoidance of the nighttime dosing. However, they are also having certain limitations. There are certain unavoidable fluctuations of the drug concentrations because of the poor drug designing and a poor in vitro in vivo correlation. Then there could be typical peak valley plasma concentration time profile, which might not be acceptable in certain situations. Parenteral control drug delivery system. To begin with, parenteral or parenteral comprises of the two words para, which means outside, and enteron, which is intestine. So it is a system which is devised other than, and it is besides the oral route. It is very efficient for drugs with poor bioavailability and narrow therapeutic index. It has got a very suitable specific advantage of rapid onset of action with a rapid decline of the systemic plasma levels of drug. But it has got a very salient, important disadvantage that it leads to patient discomfort. Therefore, there is an imminent need of designing a new drug delivery system which can reduce the total number of injections throughout the treatment. This approach or this concept led to the development of controlled release systems for parenteral delivery. The salient features of the parenteral controlled drug delivery system. It is important for us to know that intravenous, subcutaneous, intramuscular, intraperitoneal, and intrathecal are some of the routes through which a parenteral drug delivery can be obtained. The products which are primarily needed to be called as controlled are prepared as oil suspensions, oil solutions, and implants. The drug molecules will be released continuously from such reservoirs after being injected at a rate determined by the characteristics of each formulation. This continuous release of drug molecules will result in a prolonged blood drug level. The rate of absorption and hence the duration of therapeutic activities will be definitely determined by the nature of the vehicle, which becomes an important selection criteria. It has been just mentioned that the parenteral route per se is very painful, and the frequency of injections, if more, will probably result in patient non-compliance. Therefore, there is an imminent need for developing a controlled drug delivery system for the parenteral route as well. In order to probably meet this goal, there have been a variety of formulation approaches which have been used by different manufacturing scientists. The routes which are commonly exploited include, number one, the preparations which are viscous and are usually made by using water-miscible vehicles and sometimes make use of the aqueous solution of gelatin. The sheer viscosity perhaps helps in a controlled drug delivery. It can also use water-immiscible vehicles like the vegetable oils along with the water-repelling agents like use of aluminum monostearate. Thixotropic suspensions can be made into parenteral drug delivery systems. Water-soluble drug derivatives, complexes, salts and esters can be exploited. Dispersion of microspheres or microcapsules using polymers which are biodegradable in nature, including lactide, glycolide, homopolymers or copolymers can also be used. Then there can be an approach where a co-administration of vasoconstrictors can release or help the release of a drug as per the desire of prolonged controlled release through the parenteral route. Another very suitable approach for getting a controlled drug delivery via the parenteral route is using the concept of implantation. There can be a variety of implantable parenteral controlled drug delivery devices, and they can be classified as the first one, controlled drug release by diffusion mechanism showing devices. So we can have under this the membrane permeation controlled drug delivery systems, wherein they can use a non-porous membrane, a porous membrane or a semi-porous membrane. The second variety could be the matrix diffusion controlled drug delivery, which can make use of the lipophilic polymers, hydrophilic polymers and the porous polymers. Additionally, we can have micro-reservoir dissolution controlled drug delivery, which might use hydrophilic reservoir in a lipophilic matrix and a reverse lipophilic reservoir in hydrophilic matrix. Then we primarily can also have membrane matrix hybrid type drug delivery system, which primarily has the same concept as earlier, You have the lipophilic membrane with a hydrophilic matrix and a hydrophilic membrane with a lipophilic matrix. The second variety of implantable parenteral controlled drug delivery systems are usually the one which require some type of activation. Therefore, they are called as controlled drug release by activation mechanism based drug delivery systems. It may include osmotic pressure activated drug delivery, vapor pressure activated drug delivery, magnetically activated drug delivery, ultrasound activated drug delivery, and hydrolysis activated drug delivery. The word activated primarily indicates very clearly the tool through which this mechanism has to be triggered in order for the controlled release of the drug. The screen in front of you shows a few examples of such long-lasting parenteral systems. We have the long-acting penicillin injection. Similarly, we can have a long-acting vitamin B12 injection and long-acting insulin injection, which comes by the name of Lantus and Solostar, and we can also have very similarly designed long-acting steroid injections. After understanding the parenteral controlled drug delivery system, we now have something referring to as transdermal controlled drug delivery system. The most significantly popular design in transdermal drug delivery is a transdermal patch. A transdermal patch or a skin patch is a medicated adhesive patch that is placed onto the skin to deliver a specific dose of medication through the skin and into the bloodstream. The main components which can be visualized in the screen in front of you includes the liner which protects the patch during the storage. The liner is removed prior to use. Then you will have the drug in the system, which is the drug solution in the direct contact with the release liner. Then it is having the adhesive, which serves to adhere the components of the patch together along with the adhering onto the skin. Then we have a rate controlling membrane, which controls the release of the drug from the reservoir and the multi-layer patch. And finally, we have the backing layer, which protects the patch from the outer environment. One of the most popular controlled drug delivery system is the one which probably is referred to as the transdermal drug delivery system. It has got certain very important advantages. The first is the steady permeation of the drug, allowing a constant serum drug level. If at all toxicity occurs, the effects can be diluted by simply removing the patch. The third conspicuous advantage, an alternative system for the patients who cannot tolerate oral dosage forms. So this comes very handy for those types of patients who are absolutely not able to take any drug orally. Then it is the best option for the patients who feel nauseated or are unconscious. The certain other advantages which could be highlighted are the drugs which cause the gastrointestinal upsets are suitable for the transdermal drug delivery system because it will avoid the first pass metabolism and it will not allow the drug to come in direct contact with the GI tract. The patient compliance is there due to the less dosing frequencies and a simple dosing regimen. You simply take a patch, put it up on the skin as per the instructions of the doctor, and finally remove when you think or the doctor suggests that it has to be removed. Then of course, it will also avoid the peaks and valley plasma levels. It also allows continued administration of drugs with shorter biological half-lives. My dear students, the transdermal patches is a very popular system of drug delivery. In front of you on your screens, you have different varieties or types of transdermal patches. The naming of the patches is based on the mechanism or the components which have been used. So the first one which we have is a polymer membrane permeation controlled TDDS, that is transdermal drug delivery system. The second one from top to bottom is the microreservoir controlled transdermal drug delivery system. Then we have an adhesive diffusion controlled transdermal drug delivery system, and similarly at the end we are having matrix diffusion controlled transdermal drug delivery system. The names primarily give a complete idea about the mechanism through which the drug is being impregnated onto the membranes, and finally the means and mode and the ways by which the drug enters into the skin. Let us just very quickly peruse through the steps which are involved in the transport of drug from the transdermal patch onto the skin. The first primary step is the release of drug from the drug reservoir into the rate-limiting membrane, followed by the diffusion of drug from the rate-controlling membrane through the stratum corneum, followed by sorption by the stratum corneum and then penetration through the viable epidermis, followed by uptake of the drug by the capillary network in the dermal papillary tissue, and finally the therapeutic effect on the target organ. Ophthalmic drug delivery systems based on the controlled delivery mechanism. It is a very common knowledge that there are many ailments which affect the eye, which might even lead to the loss of eyesight. In order to maintain the eye diseases, there are many ophthalmic drug delivery systems which are available. It includes both conventional as well as the non-conventional drug delivery system. Most commonly available ophthalmic preparations include the eye drops and the ointments, and about 70% of the eye dosage formulations in the market comprises these units. But these preparations, when instilled into the cul-de-sac, are rapidly drained away from the ocular cavity due to the tear flow and the lacrimal nasal drainage. Only a very small amount is available for its therapeutic effect, resulting in frequent dosing. To overcome such problems, newer pharmaceutical ophthalmic formulations such as in situ gel, nanoparticle systems, liposomes, nanosuspensions, microemulsions, iontophoretic devices, and ocular inserts have been developed. And in the last 3 decades, there has been devices which has helped to increase the bioavailability of the drug in a sustained and controlled manner. In front onto your screens, you can see a schema showing the various types of ophthalmic controlled drug delivery systems. It also shows the conventional systems, which include the solution, suspension, emulsion, ointment, inserts, and gels. Then we have the vesicular ophthalmic systems, which are the most novel of them, and they include the liposomes, neosomes, discosomes, pharmacosomes. Then we have a system which is called as particulate drug delivery system, which is targeted for ocular delivery, and it includes the microparticles and nanoparticles. Under the controlled release systems, we have got a plethora of examples, which includes the ophthalmic implants, the hydrogels, dendrimers, iontophoresis, collagen shields, polymeric solutions, contact lenses, cyclodextrins, microneedles, microemulsions, and nanosuspensions. We have a primarily advanced systems as well, which are referred to as the scleral plugs, something based on the gene delivery, siRNA, and stem cells. Students, let us now dwell upon into the advantages of the ocular drug delivery systems. The first and the primary advantage is it probably leads to an increased accurate dosing, and of course this would overcome the side effects. It provides a controlled or sustained delivery of the drug. It increases the ocular bioavailability by increasing the corneal contact along with the prolonged time. It provides targeting within the ocular globe. So if we talk of the ocular globe, then probably the drug is required to be present in this globe for a prolonged duration of time, particularly in diseases like glaucoma. It avoids the protective barriers like the lacrimation, drainage, and conjunctival absorption. It provides comfort and increased patient compliance, which is highly desirable. and it of course improves the therapeutic effect of the drug. After understanding and knowing about the various types of ophthalmic control release drug delivery systems, an important thing which is to be taken care of is the ocular pharmacokinetics. It is the same as the other pharmacokinetics of the drug through other routes. The name ocular suggests that it is the behavior of the ocular tissue in context with the drug which is delivered. It has the following salient features. The transcorneal permeation from the lacrimal fluid to the anterior chamber is to be assessed. Non-corneal drug permeation from conjunctiva and sclera to the anterior uvea. Distribution of the blood via blood aqueous barrier of the drug via blood aqueous barrier to the anterior chamber. Elimination from the anterior chamber by the aqueous humor. Turnover to the trabecular meshwork and the Schlemm's canal. Then elimination of the drug from the aqueous humor to the systemic circulation across the blood aqueous barrier. Distribution from the blood into the posterior eye across the blood retinal barrier. Intravitreal drug administration. Elimination from the vitreous via the posterior route across the blood retina barrier, and elimination from the vitreous via the anterior route to the posterior chamber. One of the other areas which probably is very often used for controlled drug delivery is the intravaginal and the intrauterine routes. The main advantages of vaginal and uterine drug delivery are mainly the avoidance of the fast-paced metabolism, gastrointestinal irritation and side effects at the gastrointestinal tract are circumvented. It is easy to administer and also provide a scope of self-administration. It has been found that the low molecular weight drugs show higher permeability through this delivery system. However, the drug delivery through this route having several drawbacks like the low bioavailability, gender specificity, cultural sensitivity, personal hygiene, local irritation, influence of sexual intercourse, and most importantly, changes of physiological conditions depending upon the age need to be considered during the design of the vaginal formulations. In order to design a suitably successful intravaginal and intrauterine controlled drug delivery devices, there are certain factors which needs to be assessed. The first factor is the physiological factor, which include the changes in the thickness of the epithelium layer, changes in the status of enzyme, monthly cyclic changes, hormones, alteration of vaginal pH, volume of vaginal fluid, and sexual arousal. The other factors include the physicochemical factors, which are the physicochemical properties of the drugs and the polymers, excipients like lipophilicity, their ionization, molecular weight, surface charge, and chemical nature. Additionally, the drug transport has to be taken into consideration, whether it is transcellularly via the concentration dependent diffusion through the cell, or it is paracellularly, which is mediated via the tight junctions, or it is vesicular or receptor mediated transport. Keeping in mind all these factors will be very highly suitable for designing a successful intravaginal and intrauterine controlled release drug delivery system. There are various types of intravaginal devices. The first one is referred to as the vaginal ring, which is shown on the screen in front of you. It can be either a sandwich type or a reservoir type, depending upon the arrangements of the polymeric membrane release layers and the drug presence. And the other are the mucoadhesive systems. And as the word mucoadhesive suggests, such systems will have an additional quality of adhering to the intravaginal membrane so that the retention time is much more enhanced. Then similarly, we have the various types of intrauterine devices which could either be non-medicated or which could be medicated. They can be copper bearing, an example is that of the copper T used as a contraceptive, and they can also be hormone releasing, which can be highly beneficial for postmenopausal women. Problems associated with intrauterine devices. Although this system is highly suitable, but it is gender specific, and because of gender specificity, it has got certain very peculiar problems associated. The first one is the menstrual problems. Almost 12% of women have to remove the copper T380A IUD device because of the increased menstrual bleeding or cramping. Then it might lead to perforation. 1 out of every 1,000 women, the intrauterine devices either get stuck or puncture the uterus, and this usually occurs during insertion. Sometimes the expulsion becomes a huge problem. Around 2 to 10% IUDs are expelled from the uterus in first few months of use if inserted right after the childbirth or in a nulliparous woman. In conclusion, what we can say is that the designing of the intravaginal or intrauterine devices with the intention of a controlled release of drug has to take a multifactorial situation into account and then devise a system which is suited for this unique environment of drug release. Dear students, let us try to conclude what we have learned from this module. The first aspect which we probably deliberated in this module was that what are the controlled drug delivery system, and of course we emphasized upon an important parameter, that is the drug release pattern from the controlled drug delivery systems. The second important thing which we probably assessed was that we are able to understand the difference between the release pattern of a controlled release drug delivery system and a conventional release drug delivery system. Also, we are able to appreciate that what are the advantages of controlled drug delivery system in context with the release profile. We are able to appreciate as well that the controlled drug delivery system will give us a flat or the plateau form of drug release. We probably will be having the drug for a longer duration of time, not only simply prolonged, we are able to have the reproducible results. The second important thing which we deliberated upon in this presentation or in this module was the various types of drug delivery systems which are focused on releasing the drug through different routes of administration. What we could conclude was that we have got a variety of controlled drug delivery systems which can release the drug at different target organs. If we want a drug to be released through the skin, we can design the transdermal drug delivery system. For example, if we want a sustained release duration of a drug for treatment of some vaginal infection, so we can have an intravaginal drug delivery system. We probably can conclude that the concept of controlled drug delivery can be very successfully used in designing a variety of dosage forms, a variety of drug delivery systems with a primary focus of bringing in the best possible patient compliance at an economical price.